Ketosis is often described as simply burning fat instead of glucose, but that leaves out the most important part of the story. You can eat enormous amounts of fat and still produce very few ketones if the hormonal signal is wrong. And one hormone acts as the dominant brake on the entire process: insulin. So what exactly is ketosis? How does the liver make ketones? Why do we make 3 different ketone bodies? And what actually causes the body to shift from glucose metabolism towards ketosis? Today, we're going to build the entire process from the ground up. Welcome back to Cancers and Mitochondrial Metabolic Disease Basics. Let's get into it. So, as alluded to in the title and in the intro, we're going to be figuring out together what is ketosis. So let's start with looking at the cell and where this process is happening at. So this is a schematic of a eukaryotic or a human cell, and we're going to be looking at this organelle right here, this mitochondrial oblong double membrane organelle. And we're going to take that cell and we're going to zoom in, and this is what it looks like in 3 dimensions, and we'll shortly look at what it looks like in 2 dimensions. But before we go there, let's first define some key terms. Ketogenesis, that's the making of ketones by primarily the liver. There is ketosis, which means that you have circulating ketones in the bloodstream. And then ketolysis is actually the utilization or the breakdown of those ketones for energy. And then a couple other important terms to define are the 3 ketone bodies. There is acetoacetate. That's the first ketone that is made in the process of ketogenesis. There is beta hydroxybutyrate, or BHB, and that is the dominant circulating ketone. It's roughly 3 to 6 times higher in concentration compared to that of the other ketones, specifically acetoacetate. And it depends on the physiologic conditions, what that ratio may be. And then acetone is a spontaneous breakdown product, much of which is inhaled. And so let's take a look at now that mitochondria and the cell from a 2-dimensional perspective, from the lens of metabolism, how we break down food into usable cellular energy. So this is the mitochondria here. It's blown up for effects. It's not this big in real life. And there are hundreds, if not thousands, of mitochondria. And there are many mitochondria per cell. So inside the mitochondria, in the matrix, is the TCA cycle. And on the inner mitochondrial membrane is the electron transport chain, where oxidative phosphorylation happens. And that is called aerobic metabolism, because you are using oxygen with the electron transport chain to make energy in the most efficient way possible using oxphos. So the journey to get to oxphos is through varying types of breakdown of macronutrients. The macronutrients are proteins, carbohydrates, and fats. So glycolysis is how we take glucose and we turn it into the end product of that pathway called pyruvate, which then gets into the mitochondria and then goes through the TCA cycle and the electron transport chain. Beta oxidation is the breakdown of fatty acids to create shorter fatty acids that enter into the mitochondria and then go through the TCA cycle and then the electron transport chain with oxygen under aerobic conditions. And then what is not well shown on this graphic, but is much better shown on this graphic, is that the amino acids are either glucogenic, which means they basically convert into the glucose pathway, or they're ketogenic, they turn into ketones, and or they both. So examples of ketogenic amino acids are leucine and lysine. The glucogenic amino acids are alanine, arginine, aspartate, etc. And then there are some that are both, like phenylalanine, isoleucine, tyrosine, tryptophan. They are both considered glucogenic and ketogenic. They all go through either glycolysis or part of glycolysis, and they go into the TCA cycle. And in order to burn those ketones for fuel, you will need oxygen and a functioning electron transport chain. So this is kind of a gestalt slide. So the input to ketone production are fatty acids or ketogenic amino acids, and that will be undergoing a process called ketogenesis in the liver primarily. There are other organs like the kidneys will have some effects here. And then the major ketones are acetoacetate, beta hydroxybutyrate, and acetone. So I'm going to start off talking about ketosis looking at this paper here. It was published in October of 2019, titled Ketogenic Diet, a new light shining on old but gold biochemistry. And it says here, diets low in carbohydrates and proteins and enriched in fat stimulate the hepatic synthesis of ketone bodies. These molecules are used as alternative fuel for energy production in target tissues. The synthesis and utilization of ketone bodies are tightly regulated both at transcriptional and hormonal levels. The nuclear receptor peroxisome proliferator activator receptor A, PPAR alpha, currently recognized as one of the master regulators of ketogenesis, integrates nutritional signals to the activation of transcriptional networks regulating fatty acid beta oxidation and ketogenesis. And let's get into the actual meat of the paper. So this is an overview of the biochemical reactions of ketone body synthesis and utilization. Ketogenesis, starting either from fatty acid oxidation, beta oxidation, or oxidation of ketogenic amino acids, leads to the formation of ketone bodies. Three distinct molecules known as beta hydroxybutyrate, acetylacetate, and acetone. They represent circulating energy molecules during fasting or prolonged exercise. Most of the biosynthetic processes occur in the mitochondria of hepatocytes or liver cells, even though a small production may be found in other tissues like kidney epithelia, astrocytes, and enterocytes. In the initial phase of fasting, tissues rely primarily on glucose metabolism, and glycogen stored in muscle and liver is depleted first. After a prolonged fast, fatty acids are mobilized from adipose or fat tissue to the liver, and they undergo beta oxidation to produce acetyl-CoA that enters the TCA cycle. Under normal conditions, acetyl-CoA from fatty acid beta oxidation is further oxidized via the TCA. Then the reduced coenzymes NADH and FADH2 allow the production of energy by the electron transport chain in the mitochondria, coupled to ATP synthesis, oxidative phosphorylation or OXPHOS. However, TCA cycle cannot handle the large amount of acetyl-CoA derived from fatty acid beta oxidation due to a combination of factors. Low levels of insulin increase fatty acid release, and together enzymes required for ketone body synthesis and utilization. Simultaneously, diversion of oxaloacetate feeds gluconeogenesis in the liver to low activity of TCA cycle due to reduced amounts of intermediates. This increases acetyl-CoA levels, which can be used as substrate for ketone body synthesis. It later says the regulation of ketogenesis. The production of ketone bodies relies on the amount of free fatty acids available for hepatic fatty acid beta oxidation, FAO. Therefore, lipolysis in adipocytes is the first control point. This pathway is activated by beta adrenergic stimulation during fasting, whereas insulin inhibits this pathway. So now we're going to get into the hormonal control of ketosis. This is really probably one of the most important pieces of this video. So hormonal regulation. Insulin, glucagon, cortisol, catecholamines, and growth hormone are the principal hormones involved in the endocrine regulation of ketogenesis. Postprandially, or after a meal, insulin lowers ketone body concentration by complementary mechanisms. First, it inhibits lipolysis, or the breakdown of fat, by suppressing the activity of hormone-sensitive lipase, HSL, thus reducing the release of fatty acids from adipocytes and their availability for ketogenesis. Second, it differentially regulates lipogenesis, or the making of new fatty acids, and beta oxidation. It simulates dephosphorylation and activation of the rate-limiting step of de novo fatty acid synthesis, ACC, resulting in elevated malonyl-CoA levels, reduced fatty acid beta oxidation via inhibition of the CPT1, and consequently the translocation of acetyl-CoA to the mitochondrial matrix, leading to reduced ketone formation in the liver, other ketogenic tissues. So what is this functionally saying? Insulin is the main hormone, the main signal that turns off ketones. So let's look at this on a picture. So here we have a system of glucose control regulated by our endocrine system, a variety of hormones. We see insulin, we see glucagon, we see adrenaline, we see cortisol, and growth hormone. You see that the vast majority of these hormones are going to raise blood glucose. They're going to raise blood glucose through either breaking down glycogen or by stimulating gluconeogenesis. Insulin is the only hormone on this table that actually drops glucose levels. And insulin is the switch that turns ketones on or off. And we're going to look at that in further slides. So just a quick physiology again. We have high blood sugar. That tells the pancreas to turn on insulin to then take insulin from the bloodstream and put it into the cells. That lowers the blood glucose. That then inhibits ketone formation. However, if we have low blood sugar, then that will also tell the pancreas to release another hormone called glucagon, and that's going to stimulate the breakdown of glycogen and gluconeogenesis, which will raise blood glucose. So look at it like this. After eating carbohydrates and or protein, you're going to absorb those nutrients from your gut. It's going to make your blood sugar rise, and that's going to stimulate the pancreas to release insulin. That's going to lower your blood glucose, ideally, as long as you have insulin resistance, and that's going to inhibit ketone production in the liver and other tissues. However, if you're fasting or if you're eating mostly fat, that's going to keep the blood sugar low, that's going to keep insulin low, and that's going to keep ketone production activated. That means your circulating ketones, beta-hydroxybutyrate and acetoacetate, are going to rise. So basically what I'm saying is insulin is the on-off switch for ketone production. So if something raises your insulin levels, then that will shut down ketones. And if you're able to keep your insulin levels low, that will effectively turn on ketones. Let's look at this in a more practical perspective. So the blue line from this C-free graphic is blood glucose in millimoles. The red line is ketones in millimoles. You can see here that when glucose is on the higher end of this chart, that means that glucose is high, that means insulin's high, that means ketones are turned off. However, if the blood glucose drops, insulin also drops, and the ketones then are turned on to replace that energy substrate. Again, you fall off the wagon, eat something that's not ketogenic, the glucose rises, the insulin rises, the ketones are immediately turned off, and then there's going to be a stabilization if you continue to try working on the ketogenic diet. Now, the next question is, what is a ketogenic diet? I pulled it from this paper that was published in January of 2024. It's titled, Does a Ketogenic Diet Have a Place Within Diabetes Clinical Practice? If a researcher has to ask that question, I think that is sad, but the question was asked and the review of the current evidence and current controversies were presented in this paper. I basically just wanted to show what a ketogenic diet was. There are many variations of carbohydrate-limited or restricted diets ranging from moderate to very low carbohydrate diets, and a strict definition remains to be agreed upon. However, Feynman provided the most commonly used categories of carbohydrate-focused diets. A moderate carbohydrate diet is between 130 and 230 grams per day, or 26 to 45% of your daily energy, based on a 2,000 calorie diet. A low carbohydrate diet is less than 130 grams, or less than 26% of daily energy intake. And a very low carbohydrate diet, or a ketogenic diet, is 20 to 50 grams per day, or 10% or less of your daily energy intake. And it later says that ketogenic diets might be considered to be a very restrictive dietary pattern. I think it's pretty obvious. Primarily consisting of high fat, moderate, or even maybe less than moderate protein, depending on your GKI, which we'll talk about later, and a very low carbohydrate intake. It is termed ketogenic, with the overarching aim being to restrict carbohydrate intake enough to induce nutritional ketosis. Have your blood glucose drop enough that your insulin drops and the ketones turn on. This is a metabolic state which imitates the biological adaptation seen in starvation without severely limiting caloric intake, and a blood ketone level between 0.5 and 3 milligrams per deciliter. Foods such as grains, potatoes, rice, fruit, starchy vegetables, legumes, and bread are substituted for other foods sources high in fat and with an increase in protein, typically animal-based. I would say that typically it is animal-based. It doesn't technically have to be animal-based. As long as the fat is high and the protein is within the range that keeps you in ketosis, it does not have to be animal-based, but probably the publication is correct. Probably most people are on an animal-based protein sources. Now, let's look at this from a graphical perspective. So let's say this is a standard diet. We have 55% carbs, we have 30% fat, and 15% protein. Then there is a middle graphic here, which is a standard ketogenic diet, which is much higher in fat, 75%, you see that. Low carbohydrates, 5%, and 20% protein. And then there are therapeutic ketogenic diets. It's a more strict ketogenic diet, where this is an extreme example, but this is 90% fat, 5% carbohydrates, and 5% proteins. That may or may not be true for you specifically. You may not be able to tolerate 5% protein. You may need more than that. But there's kind of two ways you could go about this. You could start with a higher amount of protein and check your numbers, and we'll talk about what those numbers are later, and then keep it there because your numbers are good, or lower your protein slowly to try to titrate the protein down to see if that helps your numbers. Or you can start low. We'd want to start at least a minimum amount of protein for a person, which is going to be determined between you and your doctor, but you want to start at low, and then you can slowly titrate up to see if you can keep your numbers where you want them to be in terms of ketosis. So this is what is ketosis, and I even added a little thing at the bottom of what is a ketogenic diet and what those macros may look like. But I think the most important thing to get from this is that insulin is the clear on-off switch for ketone production. And insulin can be raised by mostly carbohydrates, and amino acids can also raise insulin, and insulin will turn off ketone production. So the key of a ketogenic diet and being in ketosis is to keep that insulin low. And that way the liver is in a state that wants to make ketones and that should make ketones. And that should help keep you in the therapeutic levels that you will want to be depending on your N=1 context. If you like videos like this, please like, share, and subscribe. And until next time.